Hi, I'm Adam Cohen from the University of Pennsylvania, and I'm joined here by Drs. Yi Lin from Mayo Clinic and Dr. Sabrina Prommersberger from University of Würzburg, and we've been presenting at the CAR T-cell session here at the Myeloma 2017. So we're going to give you a little bit of a summary of what we each talked about and then a discussion about CAR T-cells in myeloma. So Yi, why don't we start with you and tell me a little bit about the Bluebird trial that you presented and the major findings of.
Great, yes. So Mayo Clinic is one of the 9 sites that participated in the BB2121 trial with Bluebird Bio and funded by Celgene. So in this trial, we used a lentivirus construct that targets a murine-derived BCMA surface with 4-1BB intracellular signaling. And so far, we have data from the standard 3+3 dose escalation cohort. And the patients in our study received CAR T at fixed dose, ranging from 50 million to 800 million cells total. We already saw promising activities above the lowest dose, which is 150 million to 800 million. And in addition, unlike the CD19 CAR T experience with leukemias and lymphomas, we have not seen as high degree of severe cytokine release syndrome or neurotoxicities. We saw 2 cases that were reversible within 24 hours of what we call grade 3, meaning it's severe enough that patient needs to be in the ICU for management. But majority of the patients, about 71% of the patients on the study, which is 21 patients dosed so far, has had mainly mild symptoms from that. So that makes it very promising in terms of both the clinical response and the lowered severity of toxicity that we've seen so far.
That's great. And what about the durability of these responses? What's your follow-up so far? What are you seeing?
Yeah, the median follow-up at the time of data cutoff, which was May of this year, is relatively short at about 4 months. But we certainly have had some patients in the earlier dose cohort that are now over 1 year post-infusion. And so far, everybody's remained in response. So at ASH this year, in December 2017, we hope to present follow-up data in terms of longer, with the duration of response with longer follow-up. In addition, this trial is now in the expansion cohort. And so one thing I should clarify is that in the dose escalation cohort, we treated myeloma patients who had at least 50% of this BCMA expression on their myeloma cells. In the expansion cohort, we plan to treat patients with much lower levels of BCMA expression, even below 50%, even at 0% by the way that we're detecting them. And we'll find out if those patients will also respond to this CAR T treatment.
Okay, that's great. It sounds like very promising data.
Thank you.
So Sabrina, I'm going to turn to you, and I know you guys are looking at actually a different way to try to get the CAR into the T cells compared to what we're doing.
We want to get rid of the lentiviral gene transfer because it's relatively expensive to produce it in GMP quality. Right. And also it's not the safest method regarding regulatory affairs. Right. And therefore, we want to use Sleeping Beauty transposition, where you use a transposon and a second plasmid with your gene of interest, and both electroporate into your cell, and then you can express the gene because of the transposon cutting out the gene of interest and introducing it into the host genome. And this method is working quite well, especially as we are using mini circles instead of the huge plasmids. Okay. So we get a really nice rate of gene expression, and the cells are really viable. And this seems to be a good alternative to the lentiviral.
Do you think it's more efficient than lentiviral in terms of getting expression?
And especially it's much lower in costs in the production.
Right. And what target are you guys looking at primarily?
We are focusing on SLAMF7 as a target for myeloma because it's uniformly expressed. And
Right. So it's obviously a number of potential targets for myeloma, and most of the studies with data so far are with BCMA, but it's certainly not the only target. And SLAMF7 is another promising one based on some of the preclinical data as well. Excellent. And I'll just briefly talk about what we're doing at Penn. So we also have a BCMA-directed CAR. It's a fully human scFv, so a little bit different than the one derived from NCI that's used in your trial. And we're also looking in heavily pretreated relapsed refractory myeloma patients. We have reported data on our first cohort of patients, which were treated just with the CAR T cells alone without any prior lymphodepletion. Really just looking for a safety signal and any activity. And what we're finding is that we're able to make CAR cells from all of these patients, and we did see objective responses in 4 of the first 9 patients who were treated, including our first subject who's now almost 2 years out in a continued stringent complete remission. We are, however, seeing some severe cytokine release syndrome as well as some severe neurotoxicity, similar to what's been described in the CD19 CAR experience. So it looks, there may be differences between the different constructs or manufacturing techniques, and I think we'll have to wait and see. So we're going forward now with expansion cohorts using cyclophosphamide alone as lymphodepletion, and we'll be presenting an update of that data at ASH as well. And we are seeing a higher frequency of responses once we add in the lymphodepletion, so I do think that's going to be important. So what do you think the future of this is going to be? Are we going to see CAR T cells being used in myeloma patients relatively soon outside of these trials?
Well, I know that Celgene is planning to open the pivotal trial to follow up on the BB2121 trial that Bluebird is currently conducting using the same construct and the same lymphodepletion chemotherapy, which in our study is Cytoxan plus fludarabine. If the data continues to look as good as it has been in the phase 1 study, then we're certainly very hopeful that the pivotal trial will be a positive one, and so that may be able to come into clinical practice fairly soon. And I know there's certainly, in the real world, it's hard to tease out each component of CAR T treatment that can contribute to the efficacy and toxicities. As you mentioned, there's so many, parts of it in terms of with the CAR construct, how are we making them, with the lymphodepletion chemotherapy, with the patient characteristics. But I certainly think in terms of as an immunotherapy for CAR T, there's definitely a role into looking at using them earlier in treatment, perhaps when patients aren't as beat up by having had so many treatments, and so their immune system might be better. Right. So there could be a role into looking at using them earlier in the sequencing of myeloma treatment options. And in addition, I think we still need to see more data about the durability of response, what the relapse looks like, but there's certainly some role into looking at how that compares and where is that sequencing with the role of stem cell transplant, which is certainly a standard of treatment for myeloma.
Great, and I think getting to your point about using it earlier, the other trial that you discussed today in your talk was the trial from Legend Biotech that was conducted in China, where their patients had only a median of 3 or 4 prior lines of therapy. And in that study, they also saw a 100% response rate, and including a very high percentage of stringent complete responses. And so I think it does get to that point that if you're getting T cells from patients earlier in their disease course where their immune system is not as perturbed, you might actually get even better responses. And so I think I agree. This is really a very promising approach right now. We now have 2 FDA-approved products for B-cell malignancies, and hopefully myeloma is going to be the next one.
Yeah. And certainly to mention what you presented earlier too in terms of looking at other targets. It's too early yet to know what the relapse looks like, but it certainly could be very relevant if right now all the CAR T strategies that we have some clinical trial data on is targeting BCMA, right. And if the relapse is lack of BCMA expression, then some of the strategies that you're pursuing could become really relevant.
Yeah, I think the dual targeting is a very good strategy. It doesn't have to be the same CAR targeting 2 different antigens, but you can also produce cells with 2 different CARs or a mixture of T cells.
Right.
So there are different ways.
There are lots of ways to do it, but infusing 2 different populations, one that hits one target and one that hits another, I think would be interesting, and seeing how each one expands.
Yeah.